Recently, Chinese military scientists successfully edited human embryonic stem cells with a gene borrowed from a creature that can survive the vacuum of space. The organism, a tardigrade, barely half a millimeter long, yet virtually indestructible. These microscopic animals can endure 1,000 times more radiation than a human and have even come back from exposure to the hard vacuum aboard the International Space Station. Now, China wants to apply that resilience to human biology. Using CRISPR-Cas9, researchers from the Academy of Military Medical Sciences inserted the tardigrade's Dsup, damage suppressor gene, into human cells. The outcome was extraordinary. The modified cells withstood X-ray levels that would typically shred human DNA. And they didn't just survive, they replicated faster. This isn't hypothetical. It's documented in military medical sciences, and it's part of a larger push to bioengineer soldiers who can survive in post-nuclear landscapes, chemical battlegrounds, and possibly even extraterrestrial environments. Tardigrades, or water bears, are extremophiles capable of surviving desiccation, extreme heat and cold, and high levels of radiation. One of their key survival mechanisms involves a protein known as Dsup, damage suppressor, which binds to DNA and shields it from radiation-induced damage. First isolated in 2016 by Japanese researchers, Dsup has since drawn international attention for its remarkable protective properties. The protein works by forming a loose molecular coating around DNA strands, reducing fragmentation from ionizing radiation. It is not only resilient but also highly adaptable, making it a subject of growing interest for biotechnologists looking to expand the boundaries of survivable environments. China's latest research, however, takes this exploration a step further, into human genetic enhancement. In a study published in Military Medical Sciences, scientists from China's Academy of Military Medical Sciences used CRISPR-Cas9 gene editing to insert the Dsup gene into human embryonic stem cells. The results were striking. The modified cells displayed enhanced survival under X-ray radiation, tolerating doses that would ordinarily be lethal. In some cases, the cells also demonstrated accelerated proliferation. This development has implications not only for medical treatments, but for military applications where personnel might be exposed to extreme environments. Envisioning a scenario in which soldiers could survive radiation exposure or operate in biologically hazardous zones is no longer the realm of speculative fiction. It is now a subject of ongoing laboratory research. This research does not exist in isolation, a trend that has been noted in multiple US Department of Defense assessments. Beijing's broader strategy includes research into gene editing, synthetic biology, cognitive enhancement, and brain-computer interfaces. These technologies are being framed not simply as tools for innovation, but as assets in a long-term strategic competition. The integration of tardigrade DNA into human cells is being viewed not solely as a scientific curiosity, but as a possible component of a wider framework for human performance enhancement in military contexts. In 2020, US intelligence officials warned about China's ambition to dominate biotechnology, citing extensive investments in DNA sequencing, data acquisition, and synthetic genomics. Many of these efforts are dual use, intended for civilian research on paper, but easily transferable to defense. China's research is also bolstered by its expansive data collection infrastructure. Investigations by the Washington Post and other outlets have detailed how Chinese genomics companies, including BGI Group, during the COVID-19 pandemic through testing kits provided to countries worldwide. While framed as humanitarian outreach, these initiatives also allowed Chinese labs to accumulate diverse genetic data at unprecedented scale. Such data sets could be used for population-level studies, but critics argue they could also aid in the development of precision-targeted biological tools, whether for therapeutics or more controversial purposes such as ethnic-specific bioweapons. Although speculative, the rapid pace and secretive nature of China's genetic research programs has left many international observers uneasy. The Academy of Military Medical Sciences, responsible for the tardigrade gene experiments, reports directly to the Central Military Commission, the top command body of the Chinese Armed Forces. The overlap between military imperatives and scientific research raises important questions about transparency and accountability. From a scientific perspective, the long-term effects of integrating non-human genes into the human genome remain largely unknown. The risks include potential off-target mutations during the editing process, disruption of regulatory genes, and unknown immune responses to foreign proteins expressed in the human body. These issues are further complicated by the limited scope of current testing, which has so far been confined to cell cultures and preclinical stages. Ethically, this area of research occupies a grey zone. International guidelines such as the Declaration of Helsinki discourage genetic modification in ways that could affect future generations unless proven safe. While the current experiments involve embryonic stem cells in vitro, the line between research and application could narrow quickly, especially under military pressures to innovate. Moreover, the introduction of gene-enhanced individuals into combat roles would represent a significant departure from established military and legal norms. Beyond the immediate physiological effects, such modifications could alter cognitive thresholds, emotional responses, or resilience to psychological stress, factors that have downstream consequences for human rights, mental health, and social reintegration. There are also broader security implications. If any nation achieves a measurable advantage in biologically enhanced personnel, it could trigger a new kind of arms race, one not based on machinery or weapons, but on the modification of human beings themselves. In a worst-case scenario, this could create an international divide between enhanced and unenhanced populations, not unlike the early nuclear. One emerging area of focus tied to this research is the future of human adaptation in space. As international interest in lunar bases and Mars missions grows, researchers are examining whether gene editing techniques could help astronauts survive the high radiation environments beyond Earth's protective magnetosphere. This isn't just theoretical. Multiple space agencies, including NASA and China's CNSA, have expressed interest in using synthetic biology to extend mission durations and reduce health risks for long-term space travel. Historically, humanity has long pursued enhancements to push the limits of performance, whether through ancient herbal tonics, 20th century amphetamines in war zones, or modern nootropics and stimulants in elite operations. The difference now is precision. With tools like CRISPR, we're no longer guessing, we're rewriting the code directly. And in doing so, we risk normalizing a model where biology becomes just another form of military hardware, programmable, disposable, and increasingly distanced from traditional notions of identity and dignity. Legal frameworks are struggling to keep up. International law remains silent on many of the core questions surrounding human gene editing for defense purposes. If a genetically modified individual commits a war crime, who is accountable? The individual, the commanding officer, or the state that engineered them? These scenarios may sound extreme, but they are not inconceivable given the pace of scientific advancement. To its credit, the international scientific community has called for increased oversight and dialogue around dual-use research of concern, DURC. Organisations such as the World Health Organization and UNESCO have advocated for global norms to address the ethical and security dimensions of genetic engineering. However, these efforts often lag behind the pace of technological development, especially in authoritarian states with fewer regulatory guardrails. Still, the potential upside of this research is not to be dismissed outright. The same Dsup protein that protects cells from radiation could be used to reduce side effects of cancer radiotherapy, or to improve the resilience of astronauts exposed to cosmic radiation during long-duration space missions. In this way, the military-backed research could produce valuable spillover benefits for civilian medicine and space exploration, if responsibly managed. The crucial question, however, is who defines what responsible looks like. In the current geopolitical climate, where trust between major powers is fragile and scientific competition is fierce, establishing mutual safeguards remains difficult. Without clear international agreements, the incentives for pushing biological boundaries may outweigh the caution needed to ensure those boundaries aren't irreversibly crossed. China's work on integrating tardigrade DNA into human stem cells represents both a technical milestone and a symbolic inflection point in the history of biotechnology. While the full application of these enhancements remains speculative, the foundations are being laid. Whether these experiments ultimately serve medical innovation, military ambition, or both, will depend on decisions made not only in Beijing, but in boardrooms, laboratories, and policy forums around the world. As nations continue to explore the possibilities of human modification, one truth grows clearer. 21st century power may not be space, land, or cyberspace, but the double helix itself.